Hello students, welcome to Raaonline. Our chapter of the day is strategies for enhancement of food production. That is part of plant breeding. And the question 1 related with the first of all, what is plant breeding? Simple answer. Plant breeding that is crop improvement. Crop improvement with in relation to crop improvement with respect to quality and quantity. Why? There is a population burst. So that's why the area of agriculture is quite reduced. So in that limited area, the quantity should be increased, and food quality means that is with good nutritional quality. Both are the main aim of plant breeding. And for plant breeding, in 1970, Green Revolution was started. Green Revolution, question generally asked from this one. And Green Revolution, the credit goes for this one is Norman Borlaug, American. Norman Borlaug, he was American. He was the originator of this particular program, that is Green Revolution. So Norman Borlaug, father of Green Revolution, Norman Borlaug. Whereas in India, M.S. Swaminathan. M.S. Swaminathan, father of Green Revolution in India. M.S. Swaminathan, father of Green Revolution in India. M.S. Swaminathan. M.S. Swaminathan, father of Green Revolution in India. Borlaug received Nobel Prize for that, but in peace field. He received Nobel Prize not in science field, that is in peace field. Why peace field? Because such a large population and limited resources for the agriculture. So for the survival of that particular population, he developed such technique within a limited area the production can be enhanced. Let's select the question number 1 from this section, that is select incorrect statement. Indian agricultural accounts for approximate 62%. No, it's a 33%. It is 33%. So A is wrong. During this period, this is right. Yes, right. This is right. That is, if we include wheat and rice both, in that case, the production from 11 million tons, it goes to 89.5. Then rice, semi-dwarf variety of rice, that is IR8, they anti-aging negative. Yes, right. It is also correct. Saccharum officinarum. Saccharum officinarum, originally grown in North India. No, wrong. It is grown in South India. So incorrect statement, that is incorrect statement actually. Now GDP, so definitely that is 11 to 89, right, but the first one is also wrong, the wrong one and wrong one. B, C, D not possible, only D possible. A and C incorrect, no. Yes, we can say that is wrong one and this is also wrong, A, B, D. Then final answer is A, B, D wrong, A, B, D wrong, because C is correct. They developed from the Taichung native. They have developed from the semi-dwarf variety Taichung native developed in Taiwan, right. The question number 2, that is study the following statement regarding inbreeding and select inbreeding strategies allow to desirable quality of two different. No, inbreeding mein there is no involvement of other one. Increase homozygosity, this is correct, first one is wrong. It also helps in eliminate a loss of desirable gene, contribute increase the fertility and productivity, this is wrong. So better one, that is 1 and 4 are wrong, 1 and 4 are incorrect, 1 and 4 are incorrect with this respect. Consider the following two statements. Effective germplasm correction, that is absolutely right. Genetic variety is root cause of any plant breeding program, that is also right. So both 1 and 2 are correct, and the 2 explains correct, yes, right. Question number 4, arrange the following steps of plant breeding program in correct order, chronological order. First one definitely collection of variability, first one. Then after collection, that is evaluation is done. After collection, evaluation is done. Then after evaluation, crossing is done. That is 3, 4. First start with the 3, then come to the 4th, then start with the second one. That is first one is collection, 4th is evaluation, 3 is cross hybridization is done, that is third step. Then selection of superior, and last one testing and release. This is the correct sequence, 2, 4, 3, 1, 5. So here 2, 4, 3, 1, 5. Third option is correct. So answer for the fourth question, that is third one. First one, collection of variability, that is germplasm collection, that is desirable traits either from same country or from outside. That is, a collection may be indigenous, collection may be exotic. And after that their evaluation is done, then evaluated parents are crossed, then testing, and finally release. Crucial step for the plant breeding program. Is selection and testing of superior recombinant. That is, when you start the steps of different breeding program, then you will get the answer of these all questions. a gene responsible for dwarfing, that is DZOV gene, that is from the Taiwan, This gene was reported, and this gene is responsible for the dwarfness. This gene is responsible for a dwarfness, that is DZOV gene. Similarly in wheat, gene for dwarfness in wheat is Norin 10. Norin 10 is gene for dwarfness in wheat. And dwarf varieties, because they mature comparatively earlier than longer one, tall variety, Dwarf varieties, their maturity time is comparatively short. That is, they perform vegetative growth earlier, and they perform early fruiting and flowering and fruiting. This is the use of dwarf varieties. Then resistant to leaf and stipe rust. Direct answer, that is a table in your NCERT. You will get the answer, that is Himgiri. Himgiri variety is resistant. To the leaf rust, stipe rust, and Hilburn disease. That is Himgiri. Then Pusa subhra. Pusa swarnim. The first one is Pusa swarnim. Pusa swarnim variety is a brassica variety. This is for the brassica only. This is brassica variety, a crucifer. Brassica, that is crucifer variety. It is resistant against white rust. Pusa subhra is variety of cauliflower. It is a variety of cauliflower. Pusa subhra is variety of cauliflower. It is resistant against black rust, curl blight, and black rot. These are. Then Pusa komal. Pusa komal is improved variety of cowpea. That is cowpea. And it is resistant against bacterial blight. So these are, that is informative type of question. This is informative type of question. Then come to the question number 8. Incorrectly paired. That is, check this one. That is Pusa swarnim. Pusa swarnim is variety of brassica. This is right. Pusa subhra is variety of cauliflower, right. Pusa snowball, and this is wrong. Actually, this is the cauliflower. This is cauliflower variety. Pusa snowball K1 is variety of cauliflower, not for cowpea. And Pusa sadavahar is right. So this is right, this is right, this is right, but this is wrongly matched. Hence, answer is question 8. Black rust of wheat. Rust in wheat is caused by Puccinia fungal disease. It is caused by Puccinia, and it is fungus. It is a fungus. Red rot of sugarcane is caused by Coletotrichum fulcatum. Coletotrichum fulcatum. This is also fungus. Late blight of potato. It is caused by a fungus, Phytophthora infestans. Phytophthora infestans. Phytophthora infestans, responsible for late blight of potato. Whereas black rot of crucifer is caused by bacteria. This is caused by bacteria. Hence, the answer is, which of the above are caused by fungi? The answer is now quite clear. That is 1, 2, 3 are fungal disease. Fourth one is caused by bacteria. Fourth one is caused by bacteria. And it is caused by Xanthomonas campestris, the name of bacteria responsible for black rot, that is Xanthomonas campestris. This is the name of bacteria responsible for black rot of crucifer. Now come to the next one, that is chili. Which variety of chili is resistant to chili mosaic virus, tobacco mosaic virus? The answer is Sadabahar. That is Pusa Sadabahar is chili variety. This is a chili variety. It is resistant to chili mosaic, tobacco mosaic and leaf curl all. Pusa Komal is cowpea variety. I have already mentioned. Pusa Subra is variety of cauliflower and Pusa Swarnim is variety of brassica. And that will be that they were discussed in that particular list. That is continued in breeding. The first one is right. That is inbreeding depression. That is quality. Instead of enhance, it declines. This is known as inbreeding depression. Come to the question number 12. That is Sonalika and Kalyan Sona. Sonalika and Kalyan Sona is related with the varieties. And these are the variety of wheat. Improved variety of wheat. Rice, Jaya, Ratna, number of varieties are there, and IR series is for the rice. Then come to the Moong Ween. Moong Ween is made resistant. Actually, it is made resistant to yellow virus by mutation. This is due to mutation breeding. And mutations are generally induced by gamma rays. They are used by the gamma rays are mostly used for the induction of mutation. Question number 14. Jaya and Ratna are semi-dwarf varieties of rice. These are the semi-dwarf variety of rice. Direct informative question. No need for there is any explanation is required here. Then Shakti Ratan Proteina. If plant breeding is done for the improvement of nutritional quality. If plant breeding is done for the improvement of nutritional quality, then the process is called biofortification. That is plant breeding for the improvement of nutritional quality. That is called biofortification. That is plant breeding for the improvement of nutritional quality. By any means, even by the introduction of a gene, that is genetically modified crops are also example for that. For example, golden rice, gene for the vitamin A is introduced. That's why that variety is what? That is rich in vitamin A. So such type of practices are kept under biofortification. Here Shakti, Ratan and Proteina, these are the variety of maize. These are the improved varieties of maize for the lysine content. They are rich in lysine. And which of the following species are crossed? Yes, Saccharum barberi is in South India actually. This is North Indian sugarcane, and Saccharum officinarum is South Indian, and this is better variety. Spelling should be Saccharum officinarum. O is there. So Saccharum barberi and Saccharum officinarum. These two varieties were crossed, and the new improved variety is good for high sugar, high yield, thick stem and ability to grow in the North India, because the South Indian variety is better for that, but it is not suitable for North India. That's why that South Indian sugarcane variety is crossed with the North Indian barberry, and the obtained sugarcane variety are very good for that North Indian belt also. So the answer for this question is 2. This is the wheat variety Atlas 66. Wheat are rich in starch, but this variety is rich in protein. Protein content is comparatively higher than normal wheat in Atlas 66 variety. That is good example of biofortification also. What is biofortification? I have already told that is plant breeding for the improvement of nutritional quality is termed as biofortification. Propagation of large number of plants in very short duration. This is possible only by micropropagation or tissue culture. This is possible only by micropropagation or tissue culture. Micropropagation or tissue culture. That is, a part of plant is taken out, known as explant. To take the number of parts and they are developed in the test tube. They are induced to develop into the new plant by this technique, that is known as micropropagation, and the plant part which is extracted or used for this purpose is explant. Explant, that is that part of plant which is used for the tissue culture. Then second one, application of hormone like cytokinin and auxin. Application of hormone like cytokinin and auxin. That is cytokinin and auxin. These two hormones are generally used in the tissue culture, and tissue culture is basically related with the totipotency property. Totipotency, that is ability of a single plant cell, living plant cell, ability of a single living plant cell to develop into new plant body, that is known as totipotency. So this particular property of plant cell is used in tissue culture practices. Obtained plant are very similar in genotype and phenotype, and they are similar to the parent also. That is, soma clones are obtained. Soma clones, these are called somatic clones or soma clone. Soma clone are obtained by tissue culture. So here the answer is 2, that is micropropagation. Now during tissue culture, that is meristems, they are germ free, pathogen free. That's why the second one is correct. That is recovery of healthy plant from the diseased plant. First one. Then soma clones, that is all the plants are very similar in genotype as well as phenotype. They are obtained by the tissue culture. With A, 2 is answer, and with V, 1 is answer. And here these are the two cases are similar, so we have to go for the third option also. Domestication, that is wild varieties. Wild varieties are bringing under human control, human management. And these wild varieties are fit in their environment, so they are used for the hybridization purposes also. And plant breeding, that is purposeful manipulation of plant, that is D with 3. So better one, that option number 1 is good. That is 1 with 2, B with 1, C with 4 and D with 3. 21st question. Cereals are generally deficient in lysine and tryptophan. These two amino acids are commonly not found in cereals, or sorry, they are deficient in cereal. These two are the amino acid. These two are the amino acid and their deficiency is seen in cereals. This is informative type of question. Then Abelmoschus esculentus. Its resistant variety that is Abelmoschus esculentus is crossed with the wild variety. This is a part of conventional breeding and obtained variety is Parbhani Kranti. Parbhani Kranti is result of conventional breeding. Abelmoschus esculentus is crossed with the other variety of Abelmoschus, other species of Abelmoschus. This is conventional plant breeding. That particular variety is responsible for the resistance against, that is, yellow mosaic virus. Resistance to stem borer in maize. Actually, there are wide varieties of plants in which some features are there which provide resistance against some insect. And one of the reason is low content of nitrogen, sugar and high aspartic acid. This is the result of resistant to the borer, that is resistant to the stem borer of maize. Find out the correct match. Pusa Gaurav. It is incorrect. That is, Pusa Gaurav is resistant to aphids. This is right. Pusa same to resistant to jacid. Right. Pusa Savani resistant to aphid. Wrong. It is resistant to shoot and fruit borer. Shoot and fruit. This is wrongly matched. That is third one. It is resistant to shoot and fruit borer. Shoot and fruit borer. Whereas Pusa A4 is right match. So the third, the Pusa Savani variety, it is a variety of okra, that is bhindi. It is variety of okra or bhindi that is resistant to shoot and fruit borer, not for the aphid. The final step of tissue culture. In final step, hardening is done. That is obtained plant by the tissue culture practices, they should be made fit to live in natural environment. So that's why hardening is done. Hardening process is gradual activity. That is, initial exposure is done for shorter duration and gradually this time increases. The time for exposure increases. Then when the plant is fit for the natural environment, then only it is shifted. The entire activity of making newly obtained plant by the tissue culture practices to make them fit for the live in natural environment is called hardening. What is totipotency? That is total capacity or potency of an organism, of a plant cell to develop into the entire new plant body. And what is embryogenesis? That is development of embryo from zygote. It's a natural activity. It is seen during sexual reproduction. But micropropagation, that is, new plants are raised from a part of tissue of a plant, that is micropropagation, and during micropropagation obtained embryo are called embryoid or somatic embryo. They are called embryoid, not embryo. They are called embryoid or somatic embryo. Somatic embryo. 26th. Which of the following induces the fusion of protoplasm? Fusion of protoplasm can be done by number 1, high voltage current, that is electrofusion. Number 1 way is high voltage current. This is called electrofusion. This is called electrofusion. First of all, two cells of different plants are taken and they are body cells, somatic cell. And if they are of plant cell, so first of all their cell wall are removed with the help of pectinase and cellulase enzyme. After the removal of cell wall, exposure of protoplasm is seen. That protoplasm is known as protoplast. That is, it is without wall, and these two protoplasts are induced to fuse. The one way of their fusion is high voltage or electrofusion. Second way by chemofusion. That is, chemofusion technique can also be applied. Chemofusion technique can also be applied, and for that PEG is used. The short form is PEG, polyethylene glycol. Question pre-medical. Polyethylene glycol is used for that purpose. And best example, best plant explant to raise viral free, pathogen free portion. Pathogen free portion is meristem. Pathogen free portion is meristem. Why it is pathogen free? Number 1, cells are in dividing state, and in dividing cell viruses do not develop. That is, in meristematic cell they are in state of division only, and in dividing cells viruses are not found. And second one, oxygen concentration is high. Oxygen concentration is high. Concentration of oxygen is high in meristematic cell, adverse to viruses. And second, they are in the state of division. Meristematic cells are in the state of division, and dividing cell, they don't have viruses, or viruses can't develop in them. That's why meristems are useful for the formation of viral free plants. And the last one, today's all over major food crop. All the crops are raised from their wild varieties. So best one is, that is, domestication of these varieties are done, and still it is applied in the plant breeding tactics. That's all. Thank you. Thank you. Thank you.